Welcome back to this next video in the series of lectures on the cell cycle. In the last video, I've told you that in the S phase of the cell cycle, the DNA is going to get replicated. You have got this homologous pair of the chromosomes, and you are, after the replication, making the sister chromatids. And these sister chromatids, they were actually attached to each other by a protein complex that was known as the cohesins. And these cohesins, they were made up of the proteins SMC1, the SMC3, the SCC1, and the SCC3. And the function of these cohesion protein is they were attached to the sister chromatids until the anaphase of the cell cycle, and they were actually working as a checkpoint to ensure that during the cell division, these sister chromatids are going to get they are going to be divided between the two daughter cells equally. This cohesion was also acting as a regulator of the transcription, and it was also involved in the DNA double strand break repair. And then we talked a little bit about the G2 phase of the cell cycle, and I've told you that the G2 phase was a quality control phase. By that I mean that it was actually going to see that the DNA is completely replicated. It was ensuring that no replication forks are there, which is actually an indication that the DNA have been completely replicated. Another important thing was that if there is any damage to the DNA in the G2 phase of the cell cycle, this transition checkpoint, the G2M transition checkpoint, was actually sensing it. That was actually going for the activation of the P53 protein, which was making the 14-3-3 sigma, and that was actually halting the cell cycle at the G2M transition point. Another thing was that I told you that in the G2 phase of the cell cycle, the cell is rapidly growing. It is synthesizing a lot of proteins, and it is preparing itself to go into the M phase of the cell cycle. Now let us discuss that the how this G2 phase is actually working as a preparatory phase for the M phase of the cell cycle. Now this seems to be a little bit crowded, but don't worry, you will get each and everything. Now in the G2 phase of the cell cycle, you are actually increasing the concentration of another cyclin, which is known as the cyclin B. And you all know that whenever there is a cyclin, there will be a CDK. So the partner for this cyclin B is CDK1, the cyclin-dependent kinase-1. So the cyclin B is going to interact with the CDK1, and when the cyclin B interacts with the CDK1 is going to get activated. Now the complex of the cyclin B and the CDK1, that is known as the M-cdk. During the G2 phase of the cell cycle, when you have increased the concentration of the cyclin B represented here with a yellow color, and the CDK1 represented by an orange color, the cyclin B is going to interact with the CDK1, therefore giving you the M-cdk. So far we have discussed different CDKs. In the G1 phase of the cell cycle, we are combining the cyclin D with the CDK4, and that was known as the G1 CDK. Then we had the complex of the cyclin E and the CDK2, that was actually the G1S CDK. Then we had the cyclin A combining with the CDK2, making the S CDK. Now this is another CDK, the M CDK, which is actually the complex of the CDK1 and the cyclin B. You might be thinking that if you have combined the CDK1 with the cyclin B, the CDK1 is going to get activated. Now this is not that much simple because certain other proteins, they are also acting and interacting with this CDK1 protein. One of the important protein is known as the CDK-activating kinase. For short, that is known as the CAK. Now if the name indicates, this is a kinase, and if it is going to interact with the CDK1, it is going to phosphorylate it. This is what is actually happening. When the CAK interacts with the CDK1, that is going to phosphorylate the CDK1 at specific position of the threonine 161. And this phosphorylation, this is stimulatory phosphorylation. That means that the phosphorylation of the CDK1 by the CAK is going to increase the activity of the CDK1. Now you can think that after the CAK have phosphorylated the CDK1, it is going to get activated. Not yet, because another protein which is known as the V1 protein, which is also a kinase, that is also acting on the CDK1. Now the V1 is going to phosphorylate the active site of the CDK1 at two positions. One is threonine 14 and the other one is tyrosine 15. So the V1 is actually adding two phosphate groups at two positions in the active site of the CDK1. Now, if you look at the phosphorylation by the V1, they are actually inhibitory phosphorylations. This means that the phosphate groups added by the V1 to the CDK1 is going to make the CDK1 inactive. So all of this event that is happening in the G2 phase of the cell cycle, that you are increasing the concentration of the cyclin B that is interacting with the CDK1, the CAK is phosphorylating it, the V1 is going to phosphorylating it, but as the phosphorylation by the V1 are inhibitory in nature, the complex of the cyclin B and the CDK, and hence the CDK1 that remains in the inactive state. Now, if you want to activate this M-cdk, the only problem is these inhibitory phosphate groups. So if you are able to remove these inhibitory phosphate groups from the active site of the CDK1, you can actually activate the CDK1. This is what is going to happen when you want to move from the G2 into the M phase of the cell cycle. Now, as you all know that if you are adding a phosphate group to a particular protein, you need a kinase. And if you want to remove a phosphate group from a particular protein, you need a phosphatase. So as we are interested in removing the phosphate groups from the active site of the CDK1, we will be activating a specific phosphatase. Now the specific phosphatase that is required for the removal of these inhibitory phosphates, that is the CDC25. So when the cell cycle, it moves from the G2 into the M phase of the cell cycle, this CDC25 is going to get activated. Now how this CDC25 is going to get activated? There is another kinase which is known as a polo kinase. Now this polo kinase, what it is going to do is that it is going to add a phosphate group on this CDC25. So the CDC25 is going to get phosphorylated by this polo kinase because this is a kinase protein. Now the addition of a phosphate group by the polo kinase to the CDC25 is going to activate it partially. By that I mean that you are activating a very small number of the CDC25, and this CDC25, when it is partially activated, it can remove these inhibitory phosphate groups from some of the CDK. Not all of the CDK that are present in the cell, but it can actually remove the some of the inhibitory phosphates from some of the CDK1. That means that out of the total CDK1 available in the cell, you are going to activate a very few of them. Once you have activated a few M-cdk, that means that the M-cdk, the CDK1 is now active. It can phosphorylate its substrates. Now, if you look at the substrates of the M-cdk, one of the substrates is the CDC25 itself. We have partially activated the CDC25 by the action of the polo kinase, but once the CDC25 have activated some of the CDKs, some of the CDK1, the M-cdk is going for the phosphorylation of the CDC25 itself, and once the CDC25 is also phosphorylated by the M-cdk, like first it has been phosphorylated by the polo kinase, now it has been activated by phosphorylated by the M-cdk, that is actually going to lead to the full activation of the CDC25. And when the CDC25 is fully activated, that means that it is going to remove these inhibitory phosphate groups from all of the CDK that are available in the cell, thereby activating all of the CDK1 that are there in the cell. The problem is that if you are going to remove these inhibitory phosphate groups from the CDK1 and the V1 is still active, that means that the CDC25 will be removing these inhibitory phosphate groups, but at the same time this V1 will be adding these phosphate groups again to the active side, thereby rendering the CDK1 inactive. Now to get rid of this problem, you have to inactivate this V1. Now once the M-cdk that is activated, what it is going to do is that it is also going for the phosphorylation of the V1, and the M-cdk phosphorylating the V1 is going to make this V1 inactive. And once you have inactivated the function of the V1, the CDC25 can remove the inhibitory phosphates added by the V1 into the CDK, and thereby the CDK1 will remain in the active form, and that is going to perform its function. So once you have activated the M-cdk, you are now you can say you can now move from the G2 into the M phase of the cell cycle, and the M-cdk is going to do a lot of things in the M phase of the cell cycle. We will discuss that in the next video.